digestion and urination, or urinary system. And so with that, again, our last two systems, and I always like fall semester specifically for this reason, not by coincidence, actually it is sort of coincidence, that this falls right before Thanksgiving. Digestive system. So give me a second, and this is not supposed to look like
It depended on the second one, because second one I didn't study the sensory at all.
But would you learn from this one or the second one?
Second one.
So I'm going to move through, at least in the very beginning. We're going to have a little bit of anatomy to go over. And that'll orient us well with what we're going to talk about on Tuesday, which is going to be the rest of the physiology of digestion. Give me a second, let me get situated here. All right, there we go. All right, digestive system. So what we're going to be looking at in the digestive system, we're going to look at all the functions related to digestion. The functions that we'll be looking at will be all contingent based on the organs as we move through the whole entire alimentary canal, which I'll describe on the next couple slides. So everything from ingestion, which is the intake of food, to the secretion of our enzymes, secretion of other fluids such as mucus and saliva. We'll look at digestion, the two different components of digestion where we can undergo mechanical digestion, that is the physical breakdown of food, combine that with the enzymatic reactions from the secreted enzymes in breaking down our food from that perspective. The mixing and propulsion, we'll talk about how do we propel food throughout the entire alimentary canal. And then lastly, how do we deal, or the second to last, how do we deal with the absorption of the nutrients from what we ate down to defecation of waste, to remove things that we do not absorb through the alimentary canal. So we're going to be looking from an evolutionary perspective, looking at more of the primitive or our earlier species. And here you can see our sponge. And the way sponges work essentially is to provide nutrients to these multicellular organisms. And those earlier ways was to take the intake of water. So sponges would basically absorb water. And by absorbing water, I mean it's actually taking in water through these pores. And these pores are able to move through. And any nutrients that are required by the cells of the sponge get absorbed directly into the cell. And anything that is not absorbed will make its way and it's spit right out through this osculum. So this is our primitive, one of our early primitive methods of food absorption. And then with the specialization of the digestive tract, you would see the presence of the alimentary canal. And what exactly is the alimentary canal? And you'll see this you'll see it in this slide. The alimentary canal is everything from the mouth over to the anus. And it is consistent with the external environment. As you saw when we talked about the external environment, whether it's light, sound, you name it, anything that we're interacting with, air, we're taking in things from the external environment and it's going to make its way through the inside of that particular specimen. And what we defecate out, it's going to still, again, make its way out back into the external environment. So this is our way, same way that sponges were able to take their external environment and move it through the cells, or at least around the cells. We do the same thing with the alimentary canal. And so these species, particularly the nematode, was able to take an intake of food through the mouth and store some of that through the pharynx, and it would just make its way through. And here you see the presence of the intestine where you have localized absorption of those nutrients. and then it makes its way out its anus. So the issue with the nematode is that it doesn't really have a way of storing food. It's constantly burrowing through and grabbing its nutrients, and it's passing through. Now the earthworm, down the sort of evolutionary line, you can see the earthworm still has that alimentary canal. But what we have, again, is we have our pharynx that assists in part of the digestion, but you see the emergence of a crop and a gizzard. And these allow for enhanced digestion. So you'll see that these specialized organs now secrete enzymes that assist more in the breakdown of those nutrients, so that essentially the earthworm can harvest more nutrients through its environment. And then looking at the salamander, as we again move further down the evolutionary line, you see this presence of the stomach, which allows storage of food. You see the intestine and the emergence of accessory organs, namely the liver as well as the pancreas. And I'll talk about the accessory organs and what that means. Then looking down the digestive tracts, here at the very, in each of these four images, you can see the length and the makeup of the alimentary canal. Again, everything from the mouth to the esophagus, all the way down to the anus. And here you can see our herbivores, and down here you can see our carnivores. And you can see a clear difference between the two, right? The clear difference is that herbivores have a longer tract. And that should make sense because plants take longer to break down. And so it needs to spend more time through the alimentary canal. It's got to make its way all the way through the small intestine, where we're going to see lengthened absorption. We're going to see the presence of the cecum, which allows the storage after the small intestine, and then through the large intestine. In both our insectivores as well as our carnivores, we don't spend that much time through the small intestine. And that's because breaking down meat takes place much faster than it does our plant material. Or in other words, the diet of carnivores are much more nutrient rich than that of plants. So you can take, for instance, a good example would be those huge whales. I forgot the name of those whales. I'm not much of a biology person, but great whales. No, that's great shark. Killer orca whales. Yeah, the big whale, yeah. Whatever the big whale is that comes in, it takes a bunch of plankton in. Those plankton are not very high in calories, so it definitely has to take in quite a bit. And for whatever that name of that whale would spend a long time through the digestive system in order to be able to absorb those nutrients, as opposed to those of carnivores where not much time is allocated to breaking down meat because it breaks down fast and there's a lot of nutrients that can be absorbed in a very calorically dense diet. So as I mentioned, I've been talking about the alimentary canal, again, consistent with that of the external environment. And in humans, you see the alimentary canal travels about 30 feet. It's got a long distance to travel as it makes its way through. The accessory organs, on the other hand, are organs that assist in the alimentary canal. So these would be things like the teeth, the tongue, salivary glands, liver, gallbladder, and the pancreas. The easiest way of thinking about which one would be considered the alimentary canal versus the accessory organs is what food actually travels through. Food travels through the mouth, travels through the esophagus, travels through the stomach, through the small intestine and large intestine. Those are all part of the canal, again, which is consistent with the external environment. Accessory organs, food doesn't travel through the teeth or the liver or the gallbladder or the pancreas. The gallbladder, the liver, and the pancreas dump things into the alimentary canal to assist in digestion as well as absorption. Now, looking at it in a molecular perspective, we're going to be looking at two terms that we've already seen before: catabolic reactions as well as anabolic reactions. Again, this is a throwback to our biochemical lecture where we saw the breakdown, which is catabolic reactions, taking large molecules and breaking them down to their smaller constituents. This process in our catabolic reactions is basically taking whatever you eat. And whatever you eat, if we're going to take the example of a hamburger, it contains all of the 4 organic compounds, macronutrients, our carbohydrates, our proteins, fats, as well as the DNA within those cells. And we're going to break all of those polymers down into their monomers, their individual components. So catabolic reactions are going to be able to release energy. We're releasing the energy that comes from the breakdown of our molecules. This catabolic reaction you saw in cellular respiration. Anabolic reactions are taking those smaller constituents, those monomers, and building larger molecules out of it. So if you've taken that steak and you've broken them down to their amino acids, those amino acids can then again be formed into bigger proteins. We can form them into any globulins, you can form them into hemoglobin, you can form them into any of the proteins that liver produces. You can turn any of those glucose molecules that you've just ingested, that you've broken down through catabolic reactions, and form liver or muscle glycogen for energy storage. So in this process, this is the breakdown of those macronutrients, bringing them to their smaller constituents, and then building back from it. So whenever you hear the concept of you are what you eat, no, you're not. You actually take down whatever you just ate, break them down into smaller things, and you build on top of that. Mechanical digestion. So we're going to start with defining some terms with mechanical versus chemical digestion. What exactly is mechanical digestion? It's basically the physical breakdown of food. How do we break apart that hamburger into smaller pieces? And mechanical digestion first starts off with mastication. And the fancy term for mastication, well, that is a fancy term. That is chewing. It's what we're doing when we're chewing food. We're using our teeth, the accessory organs, known as our teeth, to physically break that food apart into smaller bites. The process of swallowing, the process of mixing involves whatever you've chewed and swallowed and mixing that with enzymes in the stomach. And now you've got your food mixed together, and what better way to get it to all mixed together through what a term we're going to call peristalsis, which is going to be muscle movement. Muscle movement in the alimentary canal that allows that mixing of those enzymes, namely the gastric juices that we'll be looking at later, mixing those gastric juices with the food that was chewed and swallowed, allowing us to create this slurry of stuff, and it makes its way through the alimentary canal to further digestion as well as absorption later down down the alimentary canal. Now, chemical digestion, on the other hand, is going to involve catabolic reactions. Again, the breakdown, the enzymatic breakdown of our molecules. So in our catabolic reactions, going back to earlier, catabolic reactions involve terms that you'll see later on, hydrolysis, which is the lysing, the break apart of molecules with water. So water molecule comes in and breaks that apart, which you'll see on the back of that original slide. This results in the release of energy. So in other words, this is an exergonic reaction. Now, our endergonic reactions are those for anabolic reactions. So these are endergonic, in which we're going to require energy to build our molecules. So in the case of glycogenesis, that requires ATP. ATP that we were able to harvest from our catabolic reactions. So here you can see catabolic and anabolic reactions together is a whole process of metabolic or metabolism. For anabolic reactions, this would be called dehydration. Synthesis. In order to synthesize a glycogen molecule, we have to remove water from that process. So going back to that original slide, the breakdown of fats get broken down into fatty acids and glycerol. So in these cases, we're going to be breaking those ester linkages that link the glycerol molecule to those fatty acids. Carbohydrates, any polysaccharides, we're taking those polysaccharides and breaking them down into monosaccharides. Best case example you can see in the bottom is taking table sugar. Table sugar below, which is going to be 1 glucose and 1 fructose saccharide. These two monosaccharides that are linked together by a glycosidic bond get broken up. You have water in that process. Water comes in for hydrolysis, introduce water, and that will break apart that glycosidic bond. where you originally had an oxygen molecule bound to these two monosaccharides. That gets bound up, you break apart one, and you break apart the other. Again, releasing energy at the same time requiring a water molecule. So this is something that in the process of chemical digestion actually involves water. So you may hear at some point, and if any of you guys ever read any survival stuff, if you're out in a place where you don't have much access to water, but you have food, the process of chemical digestion, of breaking apart those food molecules that you've just taken in, require water. So can contribute to dehydration much faster. So last thing don't take that advice or anything. If you're ever in a survival situation, just remember digestion involves water. So you need that water in order to maintain normal blood pressure, as we learned in our prior section. But if that water is being used to assist in chemical digestion, well, at least you're well fed, not well hydrated. The other portion is breaking down our proteins. So again, our polypeptides get broken down into smaller peptides as well as amino acids. We might not be able to break down all these big proteins because again, they might be very long proteins, but we can certainly break them down into smaller peptides that can be absorbed down in the small intestine. Now, the next several slides are going to talk about the cross-section. And I'm going to kind of move through this for the most part so that you guys get the main idea of what's happening, but I'll trust that you guys can go through and read on your own. But through a generic anatomical cross-section of the alimentary canal, again, the canal that food travels through, in the case of the lumen would be the cavity on the inside that food travels through. You can see the different layers, and these layers we've studied quite a bit. You'll see the mucosal layer. The mucosal layer will break down each of the layers. Mucosal layer is the layer that's most exposed to the lumen. You'll see the submucosal layer, which is going to be our connective tissue surrounding. You'll see the muscularis layer, as you can surmise by the name, is going to be the muscular layer. And then the serosa adventitia, the things that are connected to all the surrounding organs. And with that, we'll take a look at the innermost layer, the mucosa. And depending on where we're looking at, we're either going to have non-keratinized stratified squamous epithelium or simple columnar epithelium. I know that sounds like a mouthful, but the first thing we can do is look at the first word. The first word is keratin. Keratin is a protein that's present in hair and nails. So it's a very tough structure. Not something that we would really want in the alimentary canal. So non-keratinized, stratified. What does stratified mean? Multiple layers. Multiple layers of squamous cells, squamous being flat cells. Epithelial cells, which is the characteristic of cells that are meant for protection. So non-keratinized stratified squamous epithelial cells, which you'll be able to see on a later slide, allows for food to travel through areas where we get high traffic. So pharynx, esophagus, as well as the anus. As food travels through, we can slough off the top layers of the epithelial cells. Namely, the top layers, which will get sloughed off, and you still have other flat cells below that still allow for protection. And you'll see that the simple squamous, and these are just a bunch of cells that are all kind of piled up together. As we slough off that top layer, and this is the lumen, and this is our cross-section. As that top layer gets sloughed off, you'll see all the basal cells below that are continually dividing to make sure that we serve protection for all the layers underneath, namely the submucosa as well as the muscularis. Now, on the other areas of the alimentary canal, you'll see the simple columnar epithelial cells. Simple meaning one layer thick, columnar just giving the shape, and again, epithelial cells. And in this case, these cells are mainly geared for secretion or absorption, depending on what area of the alimentary canal we're looking at. So again, simple layer, one cell thick, which makes sense for getting things to move across that one cell layer membrane. And where would we see this? We would see this in the stomach as well as the intestines. And then embedded within those epithelial cells, you'll start to see other glandular cells that have exocrine function. These are things that they're going to be secreting into the lumen of that alimentary canal. The layer beneath is just a bunch of connective tissue underneath. Again, we'll look at that later, where we have our capillary network that's going to feed and provide nutrients to the mucosal layer. The muscularis mucosae is a layer within the mucosa itself. This is not the muscularis that you see, which is the third layer. This is a sublayer within the mucosa. And the purpose of that mucosal layer, or the muscularis mucosae, is to provide increased surface area. So you get these folds that are made in the mucosa that allow for nutrient absorption. So what it would look like on that mucosal layer, it's basically a bunch of folds looking like this. And we'll be able to see those structures as we go through the stomach as well as the small intestine that give us this wrinkled look on the inside. So by increasing the surface area, we increase the amount of contact area that food travels through. We don't want a smooth tube flowing through. We want a bunch of ruffles in there so that it can make its way through and get absorbed as much as possible within the proximity of that lumen, or the mucosal layer. Now within the submucosa, you'll see submucosa, again, the lamina propria, which is basically the connective tissue surrounding the mucosal layer, are going to contain your arteries and veins that feed the capillaries embedded within the mucosal layer. So arteries and veins, and naturally, every time when you've done your, in anatomy, you've learned when you see arteries and veins, you're also going to see nerves. And that nerve layer is called the Meissner's plexus. Meissner's plexus. And that is going to be the parasympathetic division. These are parasympathetic pathways that come in and innervate the mucosal layer. So this is what we're doing to increase the blood flow to the mucosal layer. So again, parasympathetic pathways, just like you saw in the test. Parasympathetic pathways that allow vasodilation to increase blood flow to the mucosal layer. Now the muscularis layer, again, the outer layer on here, is basically going to consist of skeletal muscle or smooth muscle. And again, depending on where we are in the alimentary canal, you're going to either see voluntary actions or involuntary actions. Voluntary include the act of swallowing, control of defecation, where we see skeletal muscle that'll control either input or the output of food to the alimentary canal. Smooth muscle is going to involve, areas with smooth muscle are going to involve autonomic regulation. So these are going to be areas that are going to contract on their own. Involuntary control of those areas. And typically in the muscularis layer, you're going to see two different layers. And if I can just draw just a generic tube through, you're going to see these circular layers. You're going to have a circular layer, and then you're going to have a longitudinal sheet that runs along the length of the canal itself. And the purpose of this is to assist in the squeezing that takes place. We're going to squeeze segment by segment to be able to propel food down the alimentary canal. So that's going to be consistent. The intercircular muscles are going to allow contraction within that particular unit. And then the longitudinal sheets are going to create these wave-like patterns that will allow the propulsion of food through down the length of that canal. And then another thing that I don't really require you guys to know is the myenteric nerve plexus. That basically just allows innervation of the mucosal layer for the production of our chemical enzymes and so forth. So myenteric nerve plexus are things that you're going to be able to see in the stomach that allow the mucosal layer to do what it's supposed to do. Serosa and adventitia, again, the outer layer. This is going to be the outer layer that wraps around. Namely, it's going to contain serous fluid. Serous fluid is basically a lubricant. It allows for the slipperiness. Now imagine, you've just eaten something and you've got all this muscle movement taking place in the alimentary canal. So all the surrounding tissues will want to be surrounded by serous fluid to allow that slipperiness. You don't want friction happening along the organs. You want that fluid to allow those tissues to slip and slide around each other. that's all I really want to mention on that side. The next couple slides are going to talk about the peritoneum. This I don't need you guys to really focus too much on. Basically, all this is the protection of the GI tract. And in these cases, you're going to get these fatty layers that come in and protect the anterior of the body. As you know, we've got the ribs that help protect the lungs, the thoracic cavity, but nothing really protects the abdomen. And that's where we get the peritoneum that comes in and allows for the protection of that. And there's numerous folds, which I won't really get into. The biggest one of them is called the greater omentum. And the greater omentum is basically a fatty apron. It's the one that attaches right underneath the stomach and folds over down towards the bottom of the small intestines and wraps right back up and attaches to the end, attaches underneath. So here you can see the greater omentum that's kind of lifted up. This, again, the fatty apron is typically where you would see what the so-called beer belly. The beer belly is where you get most of the fat deposition that takes place in males. Okay. That's all I need to mention there. Next, looking at the phases of digestion is to look at everything that takes place from the mouth all the way down to the anus. And the phases of digestion, well, technically we're not doing anything, we're not digesting in the anus, so it's all up down to the very end where we see the small intestines. So we're going to start from the head down to the small intestines. And namely, these three different phases are named based on the organs that it contains. Cephalic, again, meaning the head. Gastric phase involving the stomach. And the intestinal phase involving the small intestines. So each of these areas are going to contribute to the different phases of digestion that takes place. And first and foremost, you can see that small intestine also involved in digestion. This is something that I've seen that people sometimes don't really think about, is that it doesn't end at the stomach. We're going to do more digestion within the small intestine. And this is actually the longest pathway in the alimentary canal, where the small intestine is going to involve digestion as well as the other function for absorption. But right now we're just talking about digestion. So what happens in the head, the cephalic phase? This is where we're going to start to involve sensory reception. We're going to receive sensory information. What information are we receiving? The smell of food, the taste of food, as well as the texture of food. So all that sensory input is going to lead into the central nervous system, and it's going to start triggering the other organs in place. Okay. So if any of you guys, I don't know if you guys eat dinner that late by the time you get home. If you're eating something, you might notice that once you start eating, you might even look at food, and you notice that your stomach starts grumbling. Why is that? Well, that's the process. When you start seeing food, you start smelling it, that's starting to prep the other organs. It's going to say, you're about to start eating some food, so start ramping up the stomach, start ramping up the small intestine, start producing your enzymes, because there's some things that we're going to start getting the intake of food taking place. Best case example is being exposed to food and salivating. The process of the production of saliva is an autonomic response. Okay? Again, is a sympathetic, or I'm sorry, not sympathetic, it is a parasympathetic activity, as you mentioned, SLUDD. Salivation is something that is parasympathetic, again, a response due to that sensory information into the central nervous system. We'll learn about what other forms of digestion are taking place in the mouth. The gastric phase, as we move from the mouth down to the stomach, we're going to learn about communication. Communication from the central nervous system, right? Our neural regulation, as well as the hormonal regulation that takes place to supplement central nervous pathways. So neural regulation is going to be if this involves the input of information, these involve the output of information. There's still some CNS sensory information taking place within the stomach, which we'll learn later on. But that is also going to trigger the ways of releasing digestive enzymes, as well as the hormones that are involved. So neural regulation involves the production of our enzymes, as well as our other digestive juices, as well as a hormone regulation, which is going to involve hormones that are going to allow communication with other organs downstream. And this, which we'll talk about later on, is called gastrin. The last phase would be the intestinal phase. And in the intestinal phase, we're moving from the stomach over to the intestines. And here you're going to learn about the two enzymes, cholecystokinin as well as secretin. Now before I get into cholecystokinin as well as secretin, I want to talk about the pH, which we'll learn much later on. The pH of saliva, again, which you'll learn later on, is about 6.5. So it's easy to say that this is a chemically neutral or a pH neutral environment. By the time we make our way into the stomach, we're going to have a very highly acidic environment where the pH is around 2. So we have a lot of hydrogen ions in this process. And we're going to learn about how we produce the acids present in the stomach. And then when we make our way into the small intestine, we're going to be back at a pH of 7. So imagine the fluid that flows from the stomach that's going to have a pH of 2 is going to get dumped into the small intestine. And we're certainly going to need some form of a buffer to prevent the pH changes, to bring it back, well, not prevent the pH changes, but to bring it back to a neutral pH. And that molecule is bicarbonate. In the same pathway that you learned about the production of bicarbonate in our red blood cells, namely through the enzyme called carbonic anhydrase, is involved, the same enzyme is involved in the production of bicarbonate. So bicarbonate in this case is going to serve to buffer the acidic chyme, which we'll learn later on, the acidic fluid that makes its way into the small intestine. At the same time, we do have accessory organs. Our accessory organs are going to be our pancreas, which is going to produce our bile, and the gallbladder that's going to be involved in the storage of bile. So those enzymes, which we'll talk about later, okay, are going to be involved in the digestion of fats. So if these organs are not actually part of the alimentary canal, and let me just draw it off key somewhere here, and I know this is not the right place for the pancreas, but as food travels through the small intestine, we need to have a hormone that's going to come in and signal the pancreas to start dumping its bile back into the small intestine. So that's where CCK comes in, cholecystokinin. And you'll learn about the origin of that name later on so that it's easier to memorize. So that pretty much sums up digestion. And we're going to learn about the pathways involved, where gastrin is going to be involved. That's going to assist in the production of our gastric juices. We'll learn about the regulation that takes place. When do we produce gastrin? And when do we stop producing gastrin? And with that, we'll take a look at the pathway of food. We move everywhere from the mouth, the pharynx, esophagus, stomach, small intestine. Small intestine is going to be divided into the duodenum, jejunum, as well as the ileum. Okay? Then we move to the large intestine, to the colon, to the rectum, to the anus. Self-explanatory. Now with that, we're going to start describing all the organs as well as the accessory organs through the alimentary canal. And as we know, alimentary canal starts at the mouth. We start at the mouth, also known as the buccal cavity, and again formed by the cheeks, as well as the hard palate, which is more anterior, and the soft palate, which is much more posterior in the back of the mouth, as well as the tongue. This cavity itself is the alimentary canal, the mouth being the alimentary canal, the cheeks, the soft palate, and the tongue being accessory organs that contribute to the cavity itself, and of which we're going to undergo two different forms of digestion, of which we illustrated earlier. Mechanical digestion, which is involving the process of mastication. We're chewing. We take food, we put it into our mouth, we use the teeth to bite the food into smaller pieces, and we're mashing that food. Not only are we biting the food, but we're using our teeth to mash the saliva in with that food that we've just ingested. And we use the lips, the muscles in the lips and the cheeks to focus that food within the teeth. So think about what your teeth are doing. I'm not saying your teeth, your tongue as well as your cheeks to focus that food directly onto the teeth to help mash that mashing to take place. So in that process, we're going to create the first step of what we call our food is that bolus of food, which is going to be what we've ingested. Combine that with saliva. We'll talk about what's in saliva on a later slide. Chemical digestion are going to involve the enzymes in this process. So we'll learn about what's composed in saliva, of which you're going to see most of it being water, about 99.5% water. Dissolved ions are going to be involved in the enzymes itself. Immunoglobulins, which are all involved in the immune system. These are things to help prevent any viruses or disease that come in, or not viruses or bacteria, that these immunoglobulins come in and attack to prevent it from entering into our body. Lysozymes are going to be, again, a bacteriolytic enzyme, basically breaking down bacteria for things that don't belong. And then the first of our enzymes are called salivary amylase. And amylase is going to be the first of our enzymes, a digestive enzyme for starch. It's what we first do to break down starch. What else is also present in saliva? We actually also have a very weak painkiller, and I keep forgetting the name of it. It's like oro something. It's a painkiller that's actually present in our saliva in very, very minute amounts. As well, combine that with immunoglobulins, sort of explains why dogs, or even cats, why they lick themselves. If you ever watch a dog lick their wound, why are they licking their wound? They're not doing it because it tastes good. They're doing it because their saliva, by instinct, contains that painkiller. It's a very weak painkiller that comes in. And then the second thing that it also does, it introduces the immunoglobulins that come in and also attack the bacteria that might be present in that wound. And the other thing that they're also doing by licking is actually removing any sort of particulate matter that might be present in the wound itself. So there is an instinctual need for animals to lick their wounds. Well, why do we not lick our wounds? Well, obviously we've got better technology than licking our own wounds. Number two, it's kind of gross. I don't know about tasting your own blood, which, I don't know, I might have an aversion to. But not only that, we also have a lot of bacteria that's also present too. So you have heard, if you ever look up cases of people that have been able to heal wounds, some of the earlier cases they've been able to use saliva for that, but also at the same time they've also had cases where you've had people get limbs amputated because of the introduction of a bacteria that was only present in the mouth as well. So don't lick your wounds. Rely on better medicine than that. Salivary glands. You'll see that we have three major salivary glands located in different areas of the mouth. And these salivary glands are tasked for the production of saliva. So we've got three glands: parotid, submandibular, and the sublingual glands. Parotid being present up at the top lateral sides of the mouth. You'll see the submandibular, which is well below the mouth. And then you've got the sublingual, which is below the tongue. How do you know these areas? If you ever bite into anything sour, you bite into a lemon, you might notice that you'll produce a lot of saliva. All of a sudden your cheeks get super watery. Why is that? You've activated your parotid glands. The parotid glands are secreting mucus. Why is that? Why does it do that? Well, think about what happens when you're biting into a lemon. It's something acidic. The pH of your saliva, as we mentioned earlier, is 6.5. And anything that comes in and lowers the pH, you're going to have an autonomic reaction where the parotid gland will dump in a bunch of watery saliva to help bring the pH back up towards 6.5. Okay? So that is, in essence, a form of negative feedback. What starts off with a drop in pH results in a bunch of water being dumped in to raise pH back up. What's characteristic of the parotid gland, as I mentioned earlier, this creates more of a watery, and let me see if I see that. I talk about that. No, I'll mention it now. This creates more of a watery saliva, where most of it's going to be water. The other portion of saliva, consists of water plus mucus. And mucus basically is a protein called mucin plus water. So introduce this mucus. Where does mucus come from? Mucus is present in saliva, but our submandibular and even more so our sublingual glands produce more of a mucousy saliva. So this sublingual gland typically is a thicker mucus, as opposed to the parotid, which is going to produce more of a watery mucus. More to come on that on a couple slides over, which I'll talk about. Other areas that also produce saliva, the lips produce a little bit of it, the cheeks, palate at the very top, as well as the tongue itself have very minor salivary glands. But the biggest contributor of those are our three at the very top. These are, just to name a few, there's actually about 1,000 other structures that actually create, or 1,000 other types of salivary glands present throughout the mouth itself. How much saliva do we end up producing? Produce about 1 to about 1.5 liters of saliva per day. That's a lot of spit. What function does it serve? Coats the oral mucosa, keeps the mouth moist. And why is that? Well, think about in our sensory lecture. Whenever we break apart food, we're releasing some of the tastings that come off food, and that dissolves in that watery environment. So we require saliva in order to dissolve those tastings so that they can make their way into the gustatory taste buds. And eventually those gustatory receptors will be able to pick up those flavors and send that over to the central nervous system. As I mentioned earlier, it's an acid buffer. So it does contain bicarbonate. So if you ever do end up eating anything sour, that comes in and that bicarbonate comes in and buffers that acid. Another thing too is soda. So those that drink soda, and I know I drink quite a bit of it. Even diet though, there's studies that show that diet soda isn't really that great for you. But soda in general contains citric acid. And citric acid is an acid and actually will lower the pH of your mouth. And so what you'll notice about soda consumption is that you produce a lot of saliva. And what comes in saliva? Within that saliva, you've got a bunch of ions in there that can start to form calcium deposits. And saliva by itself actually has something that prevents the formation of those calcium deposits. But nonetheless, those that continually drink soda will actually produce more and more and more saliva. And eventually when you exhaust the buffering capacity of saliva, that's where the pH of your mouth starts to drop and can cause the breakdown of an enamel. Any of you guys know anybody, or even you guys drink like Mountain Dew or any of the citrusy type of sodas? Nobody? Like, I don't know, Squirt, Cactus Coolers, and I don't even know about Cactus Cooler. 7-Up, for instance, Mountain Dew. Those actually have more citric acid, which is actually bad for you. And what's even worse are those that drink warm Mountain Dew. If you drink, and it doesn't have to be warm, it's just not super cold. And then even worse than that are people that sip on it throughout the day. So you're constantly, those that do that, and if you know anybody that does that, take a look at their teeth, and they've worn away the outer layer. You're taking in an acid, a warm acid. Again, what did temperature do? It increases the kinetic energy. So you've got an acid that's coming away, chipping away at that outer layer of the teeth, which we'll look at a little bit later, and eventually will cause exposure of the inner layers of teeth and teeth rotting. I know at some point, parents would have told you don't drink soda because it'll rot your teeth. They're actually right on that one. I know they spit a lot of like old wives' tales, but it actually will do that. Other things too, antimicrobial, which we mentioned earlier, tissue repair, which we mentioned what dogs do. Again, the presence of painkiller as well as other formation of, it was called, not anything you need to know, salivary, I just read this, salivary, not growth factor, salivary tooth factor, salivary something blank factor. And what that does, it allows to allow the speed up of tissue repair. So it enhances tissue repair. It's a form of growth factor which comes in and contributes to the repair of any damaged tissues. Also in saliva, contributes to digestion, called amylase. Amylase is active, which we'll see later on, is active in a pH of about 6 to 7. We'll learn later what's going to inactivate amylase. Okay, taste, which we've described earlier. Now, looking at salivary regulation, I mentioned that the production of saliva is a parasympathetic reaction. But technically, it's actually also sympathetic as well. So it's not only parasympathetic, it's also sympathetic. So I lied to you a little bit. But in parasympathetic stimulation, you produce watery saliva. So the salivary pathways from the brainstem to the medulla, all the way out to the salivary glands, produces a watery saliva. And that makes sense because what we're doing is when we introduce food into our mouth, we want to be able to mash a bunch of that food with that watery saliva. And that's going to carry all the digestive enzymes and so forth. So all these things right here, all the sensory information, stimulate the production of saliva. The touch of food, any texture that's felt by the tongue, the smell of food, the taste of food, and other psychological factors. What psychological factors are? Just thinking about food stimulates the production of saliva. If you ever think about the old experiments where we had Pavlov's dogs, and those dogs got exposed to meat powder, those dogs would salivate. It was a completely autonomic reaction. It's just they smelled food and all of a sudden they salivate. You might be doing that too if you're exposed to food as well. All of a sudden you notice that your mouth starts to get pretty filled with saliva. And again, all that is to contribute to the digestion that takes place. So what did I mean by sympathetic pathways creating saliva as well? Well, the makeup of saliva in sympathetic pathways produces much more mucus than we produce water. So in essence, the saliva becomes much thicker. Hence the sort of dry mouth that you're experiencing whenever you're under any sympathetic, whenever you're undergoing any sympathetic reaction. So if this has ever happened to you where you've had like an absolute fright of something, all of a sudden your lips get dry, your mouth gets dry, well technically what's happening is you're still producing saliva, but it's much more mucus, which gives you the feeling that your mouth is dry, when in essence, you're just producing something that absorbs that water, and that would be your mucus. And why do we do that? Well, in our sympathetic pathways, we know we're breathing a lot more. It's going to cause increased respiration and increased ventilation. And that means more air is going to be traveling through the mouth and the pharynx as well. So we know that mucus, being sticky as it is, allows the trapping of particulates that might be embedded in air. So any dust particles, any pollen, are going to stick to the mucus and not make its way into the lungs. Hence why we need a thicker mucus for that process. Now the tongue, you'll see, as I mentioned earlier, the tongue is tasked to keep food right by in between the teeth. And so that is going to be involved, at least in the mechanical digestion. The muscular action that takes place, you have the tongue, which is attached, allows moving the tongue side to side, and the intrinsic muscles within the tongue allow for the shaping of the tongue itself, so to produce the sounds that we do during speech. and then also the tongue involved in swallowing. I'm not going to spend too much time on this because that is fairly obvious. The teeth are the mouth itself. You know what a mouth looks like. You'll see all the different structures contained within. I don't really talk so much about it. The teeth itself, I don't really talk about much either. Why? Because you know teeth. For the most part, you'll see the outer layer, the inner layer, the middle layer. I don't really talk too much about it. Other than the enamel will be the outer layer, the dentin will be the middle layer, which you'll see on the next slide over. The pulp inside the tooth itself happens to be hollow. It contains the arteries, arteries, veins, and the nerves. You'll see the crown of the tooth, which is the top layer that's above the gum line. You'll see the neck, and then the root, which is going to be the root tips. Nothing else I have to describe on there as well. If you ever see the exposed yellow side, the yellow dentin, that is the broken apart enamel that's broken off, and you'll see the inner layer inside. The mouth, we have 32 adult teeth, 20 baby teeth. Next. One thing also too, is characteristic of omnivores. You have your incisors, which are sharp, meant for biting. So if you're biting into a carrot, biting into an apple, we're using the incisors to basically take a chunk out, and then you get the molars in the back. The molars have a flat surface that allow for grinding, and again, to mechanically break down our food into smaller parts, but also to allow mashing of that food with saliva as well. All right, that's it with the mouth. Well, in terms of the structures. Mastication, I won't go into more detail because I've already talked about that. This is where we're breaking down our food to create a bolus. Now let's talk about chemical digestion. We've talked about salivary amylase, which breaks down starch. So this is the first of our active enzymes. It's active in at a pH about 6.5 to 7. And by the time that amylase makes its way down into the stomach, it's deactivated or inactivated due to the acidic environment of the stomach. So thinking about an enzyme, let me just draw kind of just a random structure there. If this is what amylase looks like, and I have no idea what amylase looks like, but if amylase looks like this, what happens in the case of an acidic environment? We have an increased amount of hydrogen ions. Those hydrogen ions come in and interfere with the secondary and tertiary structures, causing denaturing of the enzyme. So now all of a sudden we get our enzyme to look like, I don't know, something like a circle or something like that, I don't know, whatever it looks like, which interferes with the active site in which starch binds to amylase. So that denaturing of the enzyme that takes place due to the acidic environment takes place in the stomach. So it's active in the mouth and inactivated into the stomach. Now, on the other hand, we have actually another enzyme, and that enzyme is called lingual lipase. So this is produced by the tongue itself. This lipase, again, looking at all of our enzymes, end in the term ACE, A-S-E. Lingual lipase is for the breakdown, salivary amylase is for the breakdown of starch, lipase is for the breakdown of fats. But it's not necessarily active in the mouth. Why? It requires a low pH. So technically, it's denatured in the mouth, and by the time it makes its way to an acidic environment, this is where lipase unravels and opens up that active site. So denaturing in this place isn't necessarily due to the hydrogen ions. It's really due to the chemical and the pH that allows for optimal the optimal operation of that particular molecule. So in this case, amylase is active in a neutral pH. Lipase requires the gastric juices in order to become active and start breaking down fats. So in sum, where do we start digesting, at least chemically digesting starches? That takes place in the mouth. Yes?
So pretty much there's no starch breakdown in the stomach, but there's
They're still, it just starts in the mouth.
Oh, okay.
What's relevant with this? Something that I actually saw was really interesting is those that practice Ramadan cannot, typically are told not to, I don't think it's the act of swallowing that's not allowed, I believe. So you're not allowed to swallow food or anything like that. But the loophole there is that you're allowed to kind of swish stuff around your mouth. So if you were to, I don't think you're allowed to drink water, but you're allowed to like swish water into your mouth, but as long as you spit it out. Something that was a poster that I went to, a long convention a long time ago, was looking at ways for people, especially marathon runners. Marathon runners that practiced Ramadan, okay? Marathon runners, if you ever look at marathon runners, they take in gels, right? So after, did anybody ever run a marathon here? Nobody? No? Okay, well anyway, not like I ever ran a marathon. But those that run marathons will eventually hit a wall. And what this so-called wall is, actually is a physiological point at which you have low blood sugar. And in order to raise your blood sugar, best way is to take in some sort of carbohydrate gel. Either you're taking in some sugar substitute of some, not sugar substitute, but you're taking in sugar some way or another to raise your blood sugar back up. But those that practice during Ramadan, during this period of fasting, are not allowed to take in food. So what they learned in this, it was a poster or it was a talk that I attended. One of the things that people that were practicing Muslim during Ramadan was that they could take in glucose. They can take in any sugary substitute, and they can swish it around their mouth. And it was okay because salivary amylase would break apart those starches, and you still absorb through the mouth. Why? Because you've got capillaries right along there. And you do absorb a very small amount, but that small amount was sufficient to keep these marathon runners running without technically ever swallowing food. So you'd ever watch anybody that was practicing during that time, they could swish sugary stuff around their mouth and spit it right back out and still get the glucose they need to raise their blood sugar back up. So we've talked about amylase as well as lipase. Let me finish up the mouth and then we'll take a break and we'll move on to the esophagus to the stomach. Process of swallowing, we're going to move our way down the pharynx. And as you saw earlier, the questions that you guys were able to answer earlier on the test about the epiglottis. Epiglottis is going to be involved, which I'll talk about in a later slide. But basically, the pharynx is going to be the next pathway for swallowing the food from the mouth. So mouth down to the pharynx, we'll see a pathway that's involved, or a process that's involved in preventing food from entering the respiratory tract. When we're swallowing, we're going to break this down to 3 different phases: the voluntary, pharyngeal, and esophageal phases. In this process of swallowing, the technical term being deglutition, you'll see the process that's taking place is the bolus of food that's present in the mouth. And the sphincter that's going to be involved, which you'll see later on, is the upper esophageal sphincter. When we talk about the esophagus, we'll talk about these. And the esophageal sphincter is right up above of the esophagus. The bolus of food that's present, and in the act of swallowing or deglutition, the first process is the soft palate, which is the palate behind the hard palate. So this particular structure right here will move upwards to block the nasal passage. So that way any air that takes place during swallowing, think about it, when you're swallowing, you're not actually inhaling air because we've occluded the nasopharynx. We can't allow any air to travel during that time. At the same time, you'll see the epiglottis. The epiglottis that's present right up here folds over and blocks the trachea to prevent food from traveling down the trachea. So this process, in order to move the soft palate as well as the epiglottis, is autonomically regulated. This is just a reflex that takes place to prevent food from traveling superiorly and to allow it to travel down the esophagus. Once we Once we start to swallow, this opens up the upper esophageal sphincter, and then once we finish swallowing, you'll see everything moves back to its original configuration. The soft palate moves back down, the epiglottis moves back up, and the sphincter closes right back up to prevent anything moving down the esophagus that shouldn't be. So breaking down these different phases, the very first phase, the voluntary phase, the act of swallowing is voluntary control. You control when you initially swallow. And in that process, what your tongue is doing is forcing that food. See how the tongue changes the shape, and it allows the food, the bolus of food to come in by the act of swallowing is going to come in and force that food to the back of the back of the throat. So this is the changing shape of the tongue, and it pushes the food, allows it to slide. That bolus of food slides back down, pinches right off at the top. Once that makes its way down to the pharynx, now it's completely involuntary. Everything from this point is autonomic. As the bolus travels down, we do have impulses from the deglutition center in the medulla. So medulla is aware that you're swallowing something in, and that's going to move the soft palate as well as the epiglottis. Now the esophageal stage is now moving its way down to the esophagus. Once the bolus enters the esophagus, you're going to see peristalsis. And again, peristalsis, if I can draw the layer of let me just draw it vertically. Here's our esophagus. We've got our circular layers, and then we have our longitudinal layers. And those longitudinal layers, combine that with our circular layers, allow the squeezing as that bolus travels. So you get that bolus of food down here, you get the squeezing that takes place that allow it to force down to the next segment. And then again, the longitudinal waves that come in and force that bolus of food down. So how long does it take for food to travel through? It takes actually fairly quickly. It's a couple seconds for that bolus of food to make its way down. Water actually moves down fairly quickly. It takes about a second to make its way down into the stomach. So if you drink water very quickly, you can feel it moving its way through, and it just enters your stomach very quickly. And that's because there's not really much peristalsis that's needed to assist water to move down. It's already slippery as is, and it doesn't rely as much on the peristaltic waves. Also, think about what happens in space. Those that are in space, where there is no gravity, how do they get food to make its way from the mouth down into the stomach? Again, peristaltic waves, which is basically just all the contractions that take place to move food down. It's like taking a toothpaste tube, squeezing it, and wringing it down towards the neck of the tube. That's basically what's happening in peristalsis. Now, the rest from the esophagus, as I mentioned, moving food down the esophagus. I really have to talk more about that because that's self-explanatory. Looking at the esophagus, as I mentioned, our histological slide, non-keratinized stratified squamous epithelium. You can see that right up at the very top. So flat cells, all stacked up, multi-layers thick. And looking at the muscularis, the layer, you have the mucosa, you have the submucosa, and the muscularis layer. Down the length of the esophagus, you'll see the first third being skeletal. You'll see the next layer being a mix of the two, and the bottom layer being smooth muscle. So there's a transition as we move down the length of the esophagus of which layers are smooth versus skeletal. The different sphincters in the esophagus, the upper esophageal sphincter, which is involved in right during swallowing, and then the lower esophageal sphincter, which is located below. The esophageal sphincter, the lower esophageal sphincter is the one that separates the stomach from that of the esophagus. And that area is right over here. This allows that bolus of food to make its way through and to make its way into the stomach. This area, though, is a common area for what's called GERD. GERD is, it's already written down for you below, gastroesophageal reflux disease, where our stomach has a pH of 2, and our esophagus is probably around a pH of about 6. So what happens with incompetence of this sphincter is that if this sphincter cannot tightly close, that causes any of the stomach contents, namely the acids and gastric juices, that can start to make its way and leak through that esophageal sphincter and can come in and irritate the sides of the esophagus. So that irritation that takes place causes the inflammation of that area, and GERD becomes an issue with a gastroesophageal reflux disease where you get those acidic stuff making its way up. Now, that inflammation that takes place is what is an issue with people that have heartburn. Another thing that's also prominent amongst those that have respiratory diseases typically also will have GERD. So any of you guys that see pulmonary patients typically will also be prescribed proton inhibitors. Proton inhibitors that would inhibit the production of protons. What protons are we talking about? Hydrogen protons, hydrogen H+. The things that contribute to the acidic nature. So if you can limit the production of acids, you can reduce the amount of inflammation due to those acids in the esophagus. So these typical drugs like omeprazole or pantoprazole typically will end in P-R-A-Z-O-L-E. These types of drugs are called proton inhibitors. So as I mentioned earlier, anybody that presents with respiratory diseases typically might actually have respiratory diseases due to GERD, right? So in those cases, stomach acids may make their way back up. They can make their way back up, and they can be aspirated, meaning they can end up inhaling some of that gastric juices into the lungs, causing inflammation of the airways. So typically anybody that presents, I don't say typically, but anybody that presents with any pulmonary diseases might also have these as well. So if you're ever looking at their medication history, you might see that on there as well.
What was the pH of 6? What is that? The pH?
pH of 6, it's still a fairly neutral environment.
No.
pH is between 6 and 6.
What is it for?
Esophagus.
Okay.
So this video just shows you what stomach is taking place, and then from that, we'll take a little bit of a break. We'll cover the stomach, and I'll get you guys out of here. I should probably connect some audio so you guys can hear something. I don't know if you really can hear the act of swallowing, but glad you can't hear it. It's probably the loudest I can get it. That's why. That's not why.
Swallowing is simple. Issues, reflexes, food entry, respiratory pathways. Nuclear contracts, which blocks the nasal passages from the lungs. The laryngeal muscles contract, closing the lungs at the top of the jaw, taking the lungs open. The epiglottis swings down on the bones of the lungs. All airways blocked off. Respiration is temporarily made. As the upper esophageal sphincter relaxes, usual contractions drive the bolus into the esophagus. The oropharyngeal stage is done, and the nose moves. During the esophageal stage, the primary wave of peristalsis, initiated by the swallowing sphincter, pushes the bolus through the
So
esophagus.
here you can see peristaltic waves,
Travels through the esophagus.
either circular muscles as well as the longitudinal muscles, lower esophageal sphincter that comes in and dumps the food in the contents of the stomach.
Goes through the esophagus. Secondary peristaltic.
Does I mentioned earlier, food takes about a couple seconds, about 4 to 5 seconds for food to make its way through, depending on how dry something is. I don't know if you guys ever try and like swallow dry toast and see how long it takes it to make its way down. No? Okay, well, don't do that. Don't do that. Don't swallow dry toast. But anything that like bread or anything that typically gets a little bit thicker, you have to rely much more on peristaltic ways to move through. But water makes its way through faster. Yeah, don't swallow dry bread. You start choking. Like Roland told me to do it. Don't do that.
Popeye's biscuit, no water.
Hmm?
Popeye's biscuit, no water.
Yeah?
Suicide.
You don't chew it? You just
No, it's still
Still dry- still dry?
Yeah.
It's so good though, no?
It is. No.
I used to demolish those whenever I get those. Which ones? Church's chicken. I don't know. It's still around, is it?
Yeah.
See, now look, but thinking about food, all of a sudden you're starting to salivate. Especially any vegans in here, you're like, oh yeah, chicken. All right. So now on to, you guys want to take a little break for a second?
Yeah.
Yeah? Okay.
Yeah.
Let's take a 5-minute break.